Hi, this video is about the role of cytokines in bronchial asthma. Bronchial asthma is an inflammatory disease of the airways, and it is associated with bronchial hyperreactivity and reversible airway obstruction. Studies indicate that T-cell derived cytokine production rather than eosinophil influx or immunoglobulin E synthesis is usually related to altered airway behavior. There is an increase in the number of CD4 Th cells in the airways, which are predominantly of the Th2 subtype. Now, the Th2 cells are characterized by secretion of Interleukin-4, Interleukin-5, Interleukin-9 and Interleukin-13. Increased expression of pro-inflammatory cytokine tissue necrosis factor alpha enhances the inflammatory process and has been linked to disease severity. Interleukin-4 is the key cytokine in asthma and is involved in the Th2 cell differentiation and immunoglobulin E production. Now, Interleukin-4 stimulates the mucus-producing cells and fibroblasts, thereby expressing the role in the pathogenesis of airway remodeling. In atopic asthmatics, Interleukin-4 induces airway eosinophilia, and this causes bronchial hyperresponsiveness or hyperreactivity. Interleukin-5 is the primary cytokine involved in production, differentiation, maturation, and activation of eosinophils. Now, it is crucial for inducing eosinophil infiltration in the airways. Interleukin-13 is present in increased amounts in asthmatic airways and has very similar biological activities to Interleukin-4. Interleukin-4 induces immunoglobulin E dependent mast cell activation, which is involved in the immediate type of allergic or hypersensitivity reactions. In the asthmatic lung, Interleukin-4 promotes cellular inflammation by inducing of vascular cell adhesion molecule, also known as VCAM1, on the vascular endothelium, which binds to integrins on the leukocytes, causing firm adhesion on the endothelium and eventually transmigrate into the interstitial space to attack pathogens. Human studies have also shown increased expression of Interleukin-9 in the bronchial biopsy sample of asthmatics. In vitro data indicates that Interleukin-9 stimulates proliferation of activated T cells, it enhances production of immunoglobulin E from B cells, and it promotes proliferation and differentiation of mast cells. Now, tissue necrosis factor alpha causes leukocyte recruitment through the upregulation of adhesion molecules and the induction of cytokine and chemokine synthesis. Elevated levels of tissue necrosis factor alpha have been detected in the sputum and bronchoalveolar lavage fluid, or also known as the BAL fluid, from asthmatics. It has the potential to stimulate fibroblasts or smooth muscle cells, indicating its role in the pathogenesis of airway remodeling. Now, Interleukin-10 is a pleiotropic cytokine. This means that it has some unanticipated effects. It is produced primarily by the monocytes. It upregulates the expression of anti-inflammatory cytokine interleukin-1A, and it suppresses the pro-inflammatory functions of cytokines like tissue necrosis factor alpha, interleukin-1B, interleukin-6, and interleukin-8. So this might have some kind of beneficial effect during airway remodeling, as it has been shown to reduce collagen synthesis and vascular smooth muscle proliferation. Interleukin-12 is the necessary cofactor for the Th1 development. It primarily regulates Th1 cell differentiation while suppressing the expression of Th2 cells. Interferon gamma is a Th1 marker. It exerts inhibitory effects on the Th2 cell differentiation. When a naive T cell encounters an antigen in the presence of antigen-presenting cells like the macrophages, dendritic cells, etc., they induce the secretion of type 2 T helper cells. Now, these activated type 2 T helper cells influence the production of cytokines like interleukin-4, interleukin-5, interleukin-9, and interleukin-13. Among these cytokines, interleukin-4, interleukin-9, and 13 binds to B lymphocytes, thereby stimulating the expression of immunoglobulin E antibodies or IgE antibodies. Now, these IgE antibodies then binds to high affinity IgE receptor, also known as the Fc epsilon receptor, on the target mast cells. This high affinity IgE receptor activates sphingosine kinase dependent calcium mobilization in the mast cells, and that leads to degranulation with the release of inflammatory mediators like histamine, prostaglandin D2, and leukotrienes, which in turn acts on the smooth muscle cells in order to induce bronchoconstriction. On the other hand, interleukin-9 act on eosinophils and thereby cause their activation, maturation, differentiation, and finally leads to tissue damage. Thanks for watching. Please subscribe to the channel and like these videos. More videos coming up. See you in the next one.